or infestation. So damage to tissue. And okay, so talking about this, I went over this. Who might be more susceptible to opportunistic pathogens? So as I was saying, like someone that's immunocompromised. So immunocompromised individuals, they might be more susceptible. And so what about for the next question? How does normal flora protect us? So by what means does normal flora, how is it capable of preventing us from like contracting infections or various diseases? Okay, excellent. So they compete with microbes that might be more damaging to us than normal. So compete with other microbes that may be more dangerous. So what are the steps to disease after exposure to pathogens? So does anyone know the first thing that happens once you're exposed by some means, some portal? Okay, excellent. So first you have adherence. So first it has to actually be able to like adhere to some kind of either like mucosal surface or something, and that's going to enable it to actually be able to enter your body. But what comes after adherence? You said invasiveness? Okay, so after that you have invasion. And so with that, once it's actually adhered, it can invade, and what would happen afterwards? Colonization, okay, good. So colonization and growth. And once it's colonized and growing in your body, then does that necessarily mean that it's causing disease, or what else do you need? So you need some virulence factors, right? So something about it is going to cause it to cause disease. And these virulence factors are going to either mediate its like toxicity, so how much damage, like what kind of damage it's going to cause, or its invasiveness. It might be able to change its surface proteins, and that might be able to make it adhere better to other parts of your body and spread. All right, so so just to give an example of a disease caused by the following portals of entry. So with a food and waterborne portal of entry, you're going to ingest something, and then it's going to cause disease by like entering your intestines and going through there. And so with that, an example of that would be like salmonella, for example. Exhalation droplets, someone like sneezes or coughs, you have viral particles floating in the air. Like that could be how a common cold would just spread, or say influenza. Direct contact. So with that, this could be like any kind of like sexually transmitted disease. That's a result of direct contact. So, just put STIs, something like that. Not necessarily just sexually transmitted disease, but that's just like an example. It could be other forms of direct contact. Indirect contact, you could maybe you touch something and then you left a viral particle there and then someone else touched it. Then they might get a disease like that. So an example would be like mononucleosis. That'll be one example. I'm just going quickly through this. Animal bites that scratch, like rabies. That's probably the first thing that comes to mind for everyone. Parenteral, that's just where you have a puncture and it's going to be injected like that, and that could pretty much be like almost anything. If you inject something directly into your bloodstream, or it goes directly into your bloodstream, then anything could really infect you at that point, if your immune system doesn't deal with it properly. And vector transmitted, so example of that would be like, say, Lyme disease, which is spread by ticks, and the tick is the vector. So by what means do most microbes invade the body? So for that, most of them are going to invade through, say, breaks in your skin, because that's the easiest way to get a disease. So breaks in skin, wounds. Another common way we get infected is by having a pathogen end up on a mucosal surface, say in your mouth, nose, something like that, and it could spread like that since the mucous membrane is very thin. So what are some structures that may physically aid pathogens in adherence and attachment? Can anyone think of something that might help a pathogen stick onto something better? For instance, Fimbriae or pili. So those are, okay, I'm not sure I spelled that correctly. Let me make sure. Okay, ignore that. Okay, yeah, Fimbriae and pili. So these are just structures outside of the cell that can help it adhere to different parts of the body, or different parts of, you have also, it's called a glycocalyx. So a glycocalyx can come in the form of, say, a capsule or a slime layer. Does anyone know the difference between the two? So a capsule is more defined, right? So the capsule is more defined, and the slime layer sort of it's easy to break apart because it's kind of more diffuse. And moving along. How are the following secreted enzymes involved in invasiveness and virulence? So hyaluronidase. This is involved in, it could break down ECM, because in ECM you have hyaluronic acid, and so this is going to help it spread throughout the skin, or not necessarily throughout the skin, but it helps it spread a lot better. So helps pathogen to spread. Okay. And coagulase. So coagulase it can be used to help clot. So it could form a clot. So say you get a cut and then you have a Staph aureus there, and this is a pathogenic one, and it can form a clot so that blood can't go there, and so it's going to protect it from any kind of immune cells that might want to go there. So This can form a clot. And the way it does that is it converts fibrinogen to fibrin, which is the active form. And fibrinolysin is just the opposite of that. This can break apart the clot. So this could be useful for pathogenic Staph aureus as well, since it can form that clot, and then it could like multiply in there, and then it can break the clot and then spread after it's gotten stronger. So this can break clots. And that converts plasminogen to plasmin. So whenever you see like that, and the ogen there, like fibrinogen and plasminogen, that means that it's the inactive form, whereas fibrin and plasmin would be the active forms of that enzyme. Okay, so lipase. Lipase, just like it sounds, is going to break down lipids. Collagenase, it's going to degrade collagen, which is a protein. Leukocidins. So these are going to be factors that are going to kill leukocytes. So your immune cells. And streptokinase and streptodornase. These ones are just like fibrinolytic enzymes. So this is an example of these ones. And lecithinase, that's going to be something that destroys red blood cells and tissue cells. So destroys red blood cells, all right. Hemolysins, similarly, it's going to lyse red blood cells. Oh yeah, sorry about that. Okay, so what are some characteristics of group A streptococci? Okay, so group A streptococci, they have the enzymes hyaluronidase. So they have hyaluronidase, and they also have collagenase, so it's obviously a very invasive species or group. And so with this, an example would be an example of something that group A streptococci could cause would be like gas gangrene. That's an example of a disease that could be caused by this group. Whoops. All right, so some characteristics of Clostridium perfringens, sorry. So with that being said, this is a pathogen that, okay, wait. I'm sorry, I got those backwards. So this is an example of Clostridium perfringens, sorry. And for group A streptococci, what you have is enzymes that break down clots, so the fibrinolytic enzymes. So because they can do that, they can, or not only can they break clots, they also have a means to inhibit clotting. So And as I was saying earlier, because they're capable of doing that, they can really like just hide themselves from the immune system, and then they can become like stronger by number, and once they then break that clot, they can spread throughout the body. All right, so LD50, that's lethal dose 50, and so what this is, it's just the dose needed to kill 50% of the animals used in the test. So the way you calculate it, say you have like LD50 of A is equal to like 50, and then LD50 of B is equal to 150. So 150 is the larger number, and what this is, it's like the lethal dose, and it's like concentration of that. So the one that's the lower concentration would be the one that is more virulent, because it takes less in order to kill 50% of the animals. So that definitely might be a question on the exam. All right, so the relationship between toxins, toxoids, and antitoxins. So toxins they're active and they can cause damage. Toxins are active and can cause damage. And then you have toxoids, which after, say, you treat a toxin with like heat or chemical treatment, they become inactive. And then an antitoxin you can get after you inject a toxoid into an animal, and then it's going to produce antitoxins, which are the antibodies for that. And you could extract those antibodies and then use them in like someone else, like say like for tetanus, you can get antibodies for that and use that if you think you've been exposed to that pathogen. All right. So the three categories of exotoxins by mechanism. So we had two different ways of distinguishing these. And so the first way we distinguished them was cytolytic, and that's similar to the cytotoxins. We'll go over the distinguishing factor of that. And so cytolytic toxins, they cause damage to the cell membranes. And that ultimately is going to lead to, as the name would imply, cell lysis and death. AB toxins, they have two components, the A and the B components. So what happens is the B component is going to like allow it to bind to a specific site, and then the A component is what's the toxic part, and that's how it's introduced, is by the B binding. B component binds specific receptor. Okay. And then the super antigen toxins. So these ones are ones that are going to cause like massive, yes. Oh, sorry about that. I think I might just like attach it then. That might help. I have two mics, so. Testing. Hello. All right. I think that sounds better. Okay, so for the super antigen toxins, it's going to stimulate a lot of immune cells, and that's going to cause like a massive inflammatory response. So recruits. All right, and then we had the cytotoxins, neurotoxins, and enterotoxins, which are 3 categories by the site of action. And with these, so cytotoxins are like the more general group. So the cytolytic toxins are a subcategory of cytotoxins. So the cytotoxins, they do the same thing. They cause cell death. And you have neurotoxins. Those are going to inhibit nerve transmission, and an example of that would be like tetanus, which is going to result in like spastic paralysis because it's going to cause your muscle to contract and you won't be able to relax it. And enterotoxins, they're going to alter the permeability of the intestinal epithelium. So that can result in like bloody diarrhea, stuff like that. All right. So with the alpha toxin, that's going to be an example of a cytotoxin. And so this is present in like Staphylococcus, or it's produced by this, so staph. And so it's going to, in eukaryotic cells, it's going to cause swelling and lysing. Diphtheria toxin, that was an example of an AB toxin. And so with diphtheria toxin, it's going to result in the ending of protein synthesis. So with that, essentially it's going to cause cell death as well. And an example of that would be like make sure I spell this right. Okay. Corynebacterium. Diphtheriae. So that would be an example of a microbe that produces that toxin. So okay, so for anthrax, also an AB toxin, but this one like consists of 3 parts. So technically you could say like A2B toxin, but for our purposes, you could just generally define it as AB. And so that's going to alter signaling pathways. And with that, it's going to ultimately cause apoptosis of the cell. And this is produced by Bacillus anthracis. So, okay. And botulinum, so botulina, I don't know if I'm pronouncing that correctly, but anyways, this toxin is, so what this is going to do is it's going to ultimately lead to a flaccid paralysis. Now, how it goes about doing that is, so first of all, the class of toxin is neurotoxin. So it causes flaccid paralysis. And so what it does is it's going to block the release of acetylcholine, which is used to make your muscles contract. And so with that, your muscles will not be able to contract, and that therefore flaccid paralysis. And tetanus toxin is just the opposite. So also neurotoxin, except for tetanus, it's going to inhibit the release It's going to inhibit the release of glycine, and glycine is used to relax your muscles. So it's going to lead to spastic paralysis. That's why whenever you hear about tetanus, you often hear that along with lockjaw, because that's when your muscles tense up and you can't relax them. All right, so for Shiga toxin. Now that's an enterotoxin. So Shiga toxin is an example of an enterotoxin, and that's going to kill your small intestinal cells, and that's going to ultimately lead to bloody diarrhea. Bloody diarrhea. And so similarly, with cholera toxin, that's also an enterotoxin. And it also falls under the category of AB toxin, but for now we'll just go over the enterotoxin part. And so that's going to also ultimately result in diarrhea and dehydration, because what happens is going to induce the secretion of chlorine ions and bicarbonate ions from mucosal cells into your intestine, and that's going to further cause water to be brought in there just because you increase or change the salt concentration. And so because of that, you're going to possibly cause dehydration. So for the lytic and lysogenic cycles of bacteriophage, so what commonly happens is the lytic cycle, and with that, the virus is going to inject its DNA into the host cell, and it doesn't get incorporated into the host genome. So what just happens is it starts producing more viral particles, and then those ultimately result in the cell lysing and releasing those. So and you have the lysogenic cycle. Now this is different in that the virus is going to incorporate its DNA into the host genome, and it doesn't necessarily cause the cell to immediately die. So it can remain dormant for a while, and after some time, like say in if you're like stressed or something, it can pop back out. And so that's similar to like cold sores with the herpes virus. It lays dormant inside your neurons, and then say you're stressed, then it could pop back out. So once that happens, once it's exposed to like a stressful environment, then it's going to enter the lytic cycle, and it's going to multiply. All right, so endotoxins. So endotoxins are, an example of an endotoxin would be like the lipid A layer of the LPS in gram-negative bacteria. And with endotoxins, they are only really released when the cell is lysed, because that's why they're like called endotoxins. They're inside like the cell wall and or inside the LPS layer, and they're not exposed to, or the host is not exposed to it until the cell lyses, and then it could release a lot of endotoxins, and that could cause inflammation and maybe even like septic shock, but generally they're not as toxic as exotoxins. All right, so that was the host microbe section. Now we're going to go over immunology and antimicrobials, which is going to be the bulk of your exam. This is going to be a really big chunk. And so to begin, what defines the virulence of a microorganism? So we went over this earlier, and so for that we talked about toxicity and invasiveness. So those are the two factors that are going to affect virulence. Invasiveness and toxicity. So obviously invasiveness, it's going to be a factor that allows the virus, the microorganism, to spread more. Toxicity is the damage that it causes. And together those can define whether, or determine whether it's going to cause disease or not, or tissue damage. So you have resistance of the host. So with resistance of the host, you have natural resistance, so that's going to be innate immunity. And you also have adaptive immunity. And we'll talk a lot about that. So why might a pathogen need to cause disease? Or, I'm sorry, what might they need to do to cause disease? So ultimately what they have to do is either they have to either neutralize threats or they have to hide themselves. So what do I mean by that when I say that? So an example would be like Staph aureus. They have something called carotenoids, and those are going to neutralize single oxygens, and those are reactive, so that's going to prevent them from getting killed by those. Another example of how they can hide themselves is also Staph aureus, as I was saying earlier. They can hide themselves by using coagulase to form that clot, and that's going to protect the immune system. Okay, so now the components of innate immunity. So you have mechanical barriers, so that's going to be like the skin. It's going to be really hard for the skin is really hard for like almost any pathogen to get through. Very few can do that. You have like mucous membranes. Those are probably going to be the ones that are most vulnerable to being invaded. You have chemical factors. So with chemical factors, you have like fatty acids, bile salts, lysozymes, complement. Those are all things that are going to be factors that are going to determine your innate immunity. Microbial factors. Microbial factors and cellular factors. So in your notes there are like a lot of examples of this, but just know these are like some of the general things. And for sake of time, I'm not going to like go through all the details of that, but we'll just leave it with these like categories. And so what are the three types of phagocytic cells involved in innate immunity, and how do they differ from one another? So in innate immunity, we have, first of all, like monocytes. And monocytes were circulating cells that are phagocytic. And if these enter like your tissue, like say you get a wound somewhere and then they're circulating your blood, they can go through the endothelial layer, and then after that they differentiate into a macrophage. And we also had neutrophils. So neutrophils are one of the granulocytes, and those are also phagocytic. So neutrophils, they're different than monocytes in that they have like a segmented nucleus. If you see it under a microscope, the monocyte has a really big nucleus, and you could definitely tell the difference. It's larger. And so these contain a lot of lysozymes too, as they are granulocytes, so they can release a lot of these lysozymes to kill cells or to kill threats. And we also had like dendritic cells. So these are also going to be found like tissue, and these ones primarily function in antigen presenting. Okay, so how do phagocytes destroy pathogens following phagocytosis? What is opsonization? What are some ways pathogens can resist phagocytosis and destruction? So one of the ways that phagocytes can destroy pathogens is after they phagocytose them and engulf them, they can have these reactive oxygen species, and these reactive oxygen species can destroy the target cells. And for opsonization, so not all phagocytes aren't able to phagocytose all cells, or all threats. So they could have some kind of factors that prevents them from being phagocytosed easily. So in opsonization, you have either complement or antibodies that are going to bind to the surface, and that's going to mark them for phagocytosis. So that's going to actually enable the phagocytes to do their job. And some ways that pathogens can resist phagocytosis and destruction. So as I was saying that not all of them can be easily phagocytosed. So one way that they could do it is, say they have like a capsule, so that can help protect them against phagocytosis. So capsule is one structure that can protect. And going back to the example of Staph aureus with its coagulase, so it can form, the coagulase can actually bind the fibrin to its surface, and then the immune cells won't be able to tell that it's actually a pathogen because its antigens are going to be hidden. Okay, so describe antigen processing in phagocytes. How does this lead to the production of antibodies? So with this, after the phagocyte actually engulfs the cell, it's going to degrade it, and then it's going to take a piece of it, and that's going to be presented on an MHC molecule. Does anyone remember which MAC class molecule it would actually be presented on in order to lead to the production of antibodies? So with to produce antibodies, it would have to be through MHC class, what? Does anyone know? 2? Okay, good. So MHC class 2. So it presents antigens following engulfment on MHC class 2. And MHC class 2 is going to bind to CD, it's going to bind to CD4 cells. So CD4 cells, and CD4 is a co-receptor. Actually, it's going to be TLRs plus CD4. So CD4 co-receptor plus TLRs. And these are present in the T helper cells. So in order for antibody production, though, specifically, we would need to activate T helper 2 cells. So CD4 plus co-receptor PTLR on Th2 cells is going to lead to B cell differentiation B cell differentiation. All right, this just turned out to be a mess. Sorry about that. All right, B cell differentiation to plasma cells. And plasma cells are the ones responsible for antibody production. All right, so describe the differences between humoral immunity and cellular immunity. What are the three characteristics of adaptive immunity? So humoral immunity is the one that's mediated by antibodies. And then we had cellular immunity. And as the name suggests, this is mediated by the cells, so specifically like T cells. So the T cytotoxic cells. Okay, so the characteristics of adaptive immunity. So for adaptive immunity, you have specificity. So you have specificity, you have memory, and tolerance. So obviously you want the adaptive immune response to be specific to whatever pathogen is present, and it also has memory so that next time you're exposed to it, you have a much faster reaction. But why might it be necessary to have tolerance? Why might you want your adaptive immune response to be tolerant? So what would happen if your immune system was like intolerant? Like, what if it just detected your own cells and then reacted to that? That would result in like an autoimmune disorder, right? So you want the adaptive immune system to be tolerant so that it doesn't react to the wrong cells or doesn't have a response to your normal cells. All right, so what T cells have CD4 and what class of MHC is CD4 associated with? So we said this earlier, and we said that T helper cells have CD4, and that associates with MHC class 2. And then for CD8, we have T cytotoxic cells have CD8 co-receptors, and these associate with MHC class 1. So that is very important to note. Definitely no difference between those two. I know you can get it's easy to get mixed up on those. All right, so next one, describe the interaction between PR and PAMP. So PR is a pathogen recognition receptor, and then PAMP is just a pathogen associated molecular pattern. So a PAMP might be something general on a pathogen that has a pattern that, say, like an LPS layer. A PR might be able to detect that one, and not specific to a specific species of bacteria, but maybe to just gram-negative that have LPS. and that allows it to have an innate response. So this is part of the innate immunity. So an example, as I was saying, would be like LPS layer is the PAMP, and then for its actual pattern that it, or for the actual receptor that detects it, it's going to be TLR4. That's just one example. Okay, so some symptoms of septic shock. So septic shock is caused by systemic inflammation, or that is what septic shock is, sorry. So it's systemic inflammation, and that can lead to things like respiratory failure, cardiac arrest. It can be pretty severe. And so something that can cause septic shock would be like, say, a gram-negative LPS layer. So if that lyses and that releases the endotoxins, which is the lipid A layer, it can lead to systemic inflammation. Oops. Whoops, I wrote that twice. Of LPS layer. Okay, so what type of immunity do extracellular pathogens tend to activate? Sorry, what was that? Okay. So for extracellular pathogens, this is going to be something that's going to be outside the cells. So say you have a bacteria invade, bacteria aren't going to enter the cell, they're going to stay outside. So that's an example of an extracellular pathogen. And if you think about it, the extracellular pathogens are the ones that get phagocytosed, and those get presented on the T helper cells, or on the antigen-presenting cells, and then the T helper cells are going to detect that. So that's going to ultimately trigger the humoral immunity. And intracellular pathogens. So with that, so you have like a virus that's going to infect inside the cell, and that cell might then present whatever is affecting it on the outside of its cell, on the MHC class 1 molecules, and a T cytotoxic cell can detect that, and because of that, it's going to cause it to kill that cell. And so that means that it's going to activate cellular immunity. So what is the precursor to T cells and B cells, and where do they mature? So T cells and B cells, the precursor to those and B cell precursor. That would be, oh, sorry. Not myeloid, lymphoid. Okay, so that would be the lymphoid precursor cells. And T cells are going to mature in the thymus. So you can just think that thymus starts with T, T cell starts with T. That's a pretty easy way to remember it. T cells mature in thymus. B cells are going to end up maturing in the bone marrow or the fetal liver. Now, monocytes and granulocytes these, the precursor to these would be the myeloid precursor. And monocytes and granulocytes are going to mature in the bone marrow. Okay. So does anyone know what it means if that acquired immunity is anamnestic? What does that mean? Like, what characteristics does the acquired immunity have? So it has to deal with memory. So anamnestic means that acquired immunity has memory. So what does that mean? That means that, say you're exposed to pathogen once, you have an immune response. The next time you're exposed to that pathogen, it's going to be a faster, stronger response. Okay. So what are some characteristics of good immunogens and poor immunogens? So what's a good immunogen? That would be something that has a high molecular weight. Because if it's too small, it's going to be very difficult in order for the immune system to even detect it. Okay, so another thing would be like it's foreign. So if it's like your own body cell, your immune system is most likely not going to react to it. Otherwise, if that happened too often, that would not be good for you. So these are like just two examples of like what a poor or what a good immunogen would be. So you could think that a poor immunogen would just be the opposite of that. So are antigens and immunogens synonymous? Is an antigen always an immunogen? So just if something's an antigen, doesn't necessarily mean it's immunogenic. So immunogen is something that's going to elicit an immune response, whereas an antigen might not do that. So an immunogen does just that. So an example of that would be heptin. So or hapten, that's something that has a low molecular weight so it's not going to be easily detectable. But then when it binds to something that has a higher molecular weight, it's going to be actually detectable. And then after that, the next time you're exposed to it, you're going to be able to react to it faster. So that's haptin, is something that's a haptin is a antigen, but not an immunogen. Okay, so the 5 major classes of immunoglobulins. So 5 major classes we had was IgA, IgG, IgM, IgE, and IgD. So the one that comprises about 80%, or is actually written incorrectly, not comprises 80% of your serum, but comprises 80% of your serum antibodies, that would be IgG. Oops. Okay. So what domains compose all immunoglobulin classes? So with immunoglobulins, you have constant domains and you have variable domains. And the constant domains are going to remain consistent throughout the different classes. So depending on what kind of constant domains you have, that's going to determine which class of immunoglobulin it is. However, the variable domains are going to determine what it's actually specific to. So constant and variable domains. And then variable domains determine specificity or specificity. Whoops, specificity. Okay. I'm having some grammar issues at the moment. All right. What is papain? And how does it react to IgG? And what are the products? So papain is just something that cleaves IgG into the two variable parts and then the constant part. So say you have like IgG like that. It's bivalent because it has 2 binding sites. And papain just is going to cleave here. And what you're going to get is you're just going to get these two, these are the 2 FAB parts. So these are the variable parts. FAB, variable domains. So this might be good if, say, you just want to have a reaction, or say you just want to use only the variable parts for some assay and you're doing an experiment. The constant part might somehow interfere. So in order to eliminate that problem, you would just cleave it with this, and then you could just use only the specific parts. So what are some major properties of the 5 major classes of immunoglobulins, and which immunoglobulin can cross the placenta, yada, yada. We'll go, so these 2 are basically the same, so we'll just go from here. So as I was saying, IgG is bivalent, and it comprises 80% of the serum antibodies. Okay, so that being said, it's also the one that can cross the placental barrier. So this is something that can ultimately lead to like hemolytic disease of the newborn if sorry, okay. So this is capable of crossing IgM is commonly present as a pentamer. So in serum, it is a pentamer. So IgG is like this, and IgM you could think of just being like that, except like 5 of those combined. And the way it works is you have like 5 of them joined on something called like a J chain. Something by J chain or joining chain. And this one is going to have low affinity but high avidity. So what that means is the bonds that it forms aren't that strong, but it forms a lot of them. So in the higher numbers are stronger. And this is what's secreted as your first line of defense, like, or this is the first one, the first antibody that gets secreted after a primary immune response. Okay, so IgA, this is an antibody that's capable of crossing, or it's one that's present in breast milk and those kind of secretions. So if a mother is breastfeeding, the baby can actually obtain the IgA antibodies from her, and that would be an example of natural passive immunity. So as far as the structure of IgA, it is present in the monomeric form, like IgG, but when it's secreted, it's actually a dimer. So secreted as dimer, so it's also connected using that J chain. And for IgE, it's found in the serum, but in very small amounts. So with IgE, it's going to actually be what's, or the antibody that's involved in like immediate type hypersensitivities or like allergies. Okay, so IgE is involved in immediate types of hypersensitivities, allergies, and that can like either be mild or it could even be like life-threatening if you go into like anaphylactic shock. So it is definitely something to worry about. And I didn't mention this earlier, but these antibodies like IgG, they have like 2 constant domains, but IgM has a 3rd constant domain. So IgM is going to have, a 4th constant domain. And Just all right. So the last antibody, was IgD. And this one is still under research, so it kind of has like its real function sort of unknown, the specifics on it are unknown. But it is present in low concentrations nonetheless. So its known function is not present, and then it is, what we do know about it is that it's going to be like abundant on the surfaces of B cells. And that's going to play a role with monomeric IgM in binding the antigen to B cells. So it's going to be especially abundant in memory cells. Okay. So what are immediate type hypersensitivities? So those are, as I was saying, allergies. And they're caused by IgE, or this is the one that's involved in, IgE is involved in type 1. The question should be about type 1 hypersensitivity. So with that, essentially what happens is the plasma cell is going to produce the IgE antibodies, and it's going to bind to the surface of a mast cell. And then that mast cell, when you have, so say this is a mast cell, and it's square, for no particular reason, but so your antibodies are going to be like that. And once you have an antigen that forms here, and it's going to form that cross-link, this mast cell, which is has all these granules, like and those are going to be like inflammatory factors or cytokines, and then so once that cross-linked is formed, it's going to release histamines, serotonin, etc., and that's going to cause inflammation, which is characteristic of a hypersensitivity. Okay, so what is the role of MHC molecules, and how do MHC class 1 and class 2 differ in their targets? So the role of MHC molecules is to present an antigen to the cells, right? So and we already mentioned earlier that MHC1 synonymous with CD8 cytotoxic T cells, MHC2 CD4 T helper cells. So when it binds to a CD8 cytotoxic T cell, it's going to lead to the T cell releasing cytokines that are going to cause lysis of that target cell. For MHC class 2, when it binds to the T helper cell, depending on if it's T helper 1, that's going to lead to, it's going to promote more phagocytosis of the macrophages by releasing the proper chemokines or cytokines for that. And it is also going to help activate that cellular immunity as well, where the T cytotoxic cells are involved. So this is like a second signal, or for the T helper 1 cell provides the second signal to the cellular immunity. Whereas Th2 cells are going to be the ones that activate our humoral immunity and stimulate the release of antibodies. Okay. So we just went over that with this. So we're just going to move on to this question now. Okay, so how are antibodies produced when antigens are introduced in the following ways? So depending on how you introduce an antigen to your body, it could result in a different mechanism for how it's actually produced or how the antibodies are released. So if it's intravenously injected, the pathway it's going to take is the antigen is going to go to your spleen, and then you're going to have the antibodies produced there. And for here, subcutaneous, intradermal, topical, or intraperitoneal, that's going to be carried by the lymphatic system to your nearest lymph node. You don't have to know the details on that because she will not be testing you on the lymphatic system, so this should be sufficient. And the mucosal surface of the mouth. So if the antigen enters that way, through that pathway, it's going to be delivered to the gullet, which lines your intestinal tract, and delivered to gullet. And once it's delivered to the gullet in your intestines, and once it does that, you're going to produce IgA, which is specific to that antigen. Okay. Describe the role of B cells in primary antibody response, what occurs in secondary antibody response. Okay, so in primary antibody response, as I was saying earlier, you have the Th2 cells. Once those get activated, they're going to, so Th2 activation leads to B cell differentiation into plasma cell. So into plasma cell and into memory cells. So if it's plasma cells produce antibodies. And the memory cells are present for a secondary immune response should you be exposed to that antigen again. So specifically in the primary immune response, you're going to be releasing IgM. So that was the pentamer. That's the antibody that's going to be part of the primary immune response, or primary antibody response. And for secondary, when you're exposed to the same pathogen or antigen, since you have those memory cells, you can have a much faster, quicker response. And in this case, you're going to have the secretion of IgG. So in primary immune response, you get IgM secretion, and secondary is IgG. All right, how does humoral immunity and cellular immunity differ and what they recognize? Okay, so we went over this a little bit earlier, but just to solidify that, humoral immunity was the one that was responsible for detecting more of extracellular pathogens. So humoral extracellular, such as bacteria, cellular, intracellular. So if they detect that something's wrong with a cell, and that could either be because it has a virus or something, then cellular immunity with the T cytotoxic cells are going to be the ones that fix that problem. Okay, so say you have an impaired humoral immunity. Well, going by this, you're going to be more susceptible to bacterial infections. If you have an impaired cellular immunity, you're going to be more susceptible to a viral infection. So you can sort of just reason through that by knowing just the basic thing here. And so a specific example of that in particular. So with humoral immunity, say you have an impaired humoral immunity, so you can't make antibodies. So an example of that would be the syndrome that's actually called, it's kind of a mouthful, but it's Burton's A gamma globulin anemia. So that's where children fail to produce antibodies. And an example of an impaired cellular immunity would be like Di-George syndrome. And that's when a child is born without a functional thymus. So remember that the T cytotoxic cells matured in the thymus. So if the child's born without a functional thymus, lo and behold, their cellular immunity is impaired. So they're going to be more susceptible to viral infections. Okay, actually. All right. What type of immunity are natural killer cells involved in? So natural killer cells, they possess a function that's similar to T cytotoxic cells. However, they are involved in innate immunity. because they're not specific. And unlike the T cytotoxic cells and the T helper cells, they don't use T cell receptors. And essentially how they function is they detect that something's wrong with a host cell. And this could be if it's like a cancerous cell or infected with like a virus, something that causes the surface proteins of that cell to change. This natural killer cell is going to release cytokines that causes that cell to undergo apoptosis or lyse. So okay. So going with active immunity. Active immunity was the one in which you are exposed to an antigen, and then you actually make those antibodies and the memory cells. So there's artificial natural immunity, artificial active immunity. And with artificial, this is when, say you just have the attenuated strain or like the inactivated form of an antigen or a pathogen injected in you, and then it's still immunogenic, however, it's not going to be pathogenic. So that's going to cause you to undergo that primary immune response. You're going to make antibodies, and you're going to make those memory cells so that next time you're exposed to it and it's like the real thing, you're not going to have no way to fight it. So that's known as vaccination. So this is vaccination. For natural immunity, natural active immunity, that's just where you're naturally exposed to pathogen, so not necessarily in activated form. You could just come down with the actual disease, but say you fight it off and you survive, then you're still gonna have those memory cells to help you fight it the next time. All right, so with the passive immunity, that's when you have cells or antibodies transferred to the actual, to the person. So with this, you're not actually having that immune response that's going to like produce all those plasma cells and antibodies. This, you're just having those antibodies directly, or the cells, plasma cells directly implanted or injected. And this could be either by artificial means or natural means. So an artificial means would be you like take, for example, as we were talking about a toxin, we inactivated it, made it a toxoid form, and we injected that toxoid into an animal, and it produced antitoxins. Take those antitoxins out, and you inject it into someone that's been exposed to the pathogen, or you think they have been exposed, and that's going to help fight that off. So say you don't have like a tetanus vaccination or something, and you think you were exposed to it, you got like stabbed or something. Well, since you don't have time to undergo that whole primary immune response and generate those plasma cells and memory cells, they're just going to put the antibodies directly in you to fight that off. So with this, so antibodies are injected, so not from a natural source, or technically it is, but like you just distinguish it from natural immunity, in that in natural passive immunity you get it through, like say breastfeeding, and that's when you have that IgA antibodies crossing, or IgA antibodies present in the breast milk. That's how you acquire them. So with that, so for attenuation, that's just where you have like a pathogenic strain, and then you make it non-pathogenic. So it's going to be inactivated by either treating it, or you can also get an attenuated strain by like cultivating in a lab for so long. Like if you cultivate it in like the bacteria you use in your microbial lab, those ones are like attenuated strains. They've been like cultivated over so many generations that they're no longer pathogenic. So you can use that. Like they could still produce an immune response, but they're not going to cause harm to you. So that's why they're perfect for vaccination, because they still produce that that primary immune response, and you generate those memory cells and the antibodies, but you don't actually risk dying to the actual disease. So inactivated pathogen. Now, it's not necessarily completely safe to use it. Like, as long as the individual is healthy, it's safe to use an attenuated strain. However, say they're like immunocompromised, then they could actually succumb to like a disease by that pathogen. All right, so nosocomial infections. These are infections that are acquired from hospital stays. So say a patient doesn't have any kind of contagious disease. Say they just have say they have something like cancer, so that's not contagious. They could potentially acquire a secondary infection, and that would be a nosocomial infection, because they are like surrounded by a whole load of microbes in the hospital, and their immune system could be compromised because of that disease that they have. And they also can undergo like some procedures that may be invasive, and that might increase their chances of actually getting a secondary infection. And these secondary infections or nosocomial infections are actually usually resistant to antibiotics. Does anyone know why? Why might a hospital-acquired infection be more resistant to an antibiotic? Yes. Okay, awesome. So, they use antibiotics every day there in the hospital. So, the more you expose bacteria to antibiotics, the more they're likely to adapt to it and evolve. So, you're going to have more resistant strains there. That's why MRSA is so prevalent in hospitals, such a big problem. So, what does an autoclave achieve? So that's a physical means of sterilization, right? So it's a physical antimicrobial agent. And specifically, it uses heat. So it uses steam heat and high pressure, about like 15 psi above normal atmospheric pressure. And that's going to enable you to get temperatures that are like 121 degrees Celsius. So that's going to be above the boiling point of water. And the significance of that is that it's capable of killing endospores. So a big problem is that oftentimes we can sterilize materials and stuff, but you can't kill the endospores because they're just so tough and robust. So that's why autoclaving is what's necessary in order for complete sterilization. Yeah, it was like 121 degrees Celsius and 200 degrees. I'm not sure which one you're referring to. Yeah, I'm not sure honestly which one you're referring to, but I would definitely check on that. So the autoclave typically operates under 121 degrees Celsius, 15 psi above atmospheric pressure. And it uses steam and heat. Okay, so some physical agents. So obviously heat is the most common one. But other agents you can use are ionizing radiation. That's an example of a physical agent. You could also use UV light. So that's something that's really common in the lab. So say you're using a cabinet, safety cabinet with that has a controlled environment. Typically after you're done working in there, you usually turn on the UV light and you leave that for a while, and that's going to hopefully kill most of the microbes. And so those are some examples of physical agents. And so how is total cell number and viable count affected by the following? So bacteriostatic agents, these are agents that are going to maintain a consistent total cell count and viable cell count. So what it essentially does is prevents growth. Prevents growth. Bactericidal agents are going to maintain a constant total cell number, but it's going to reduce the viable cell count. And bacteriolytic agents, these are going to reduce both viable cell count and total cell count. So if you look at a graph, the way it will look like is, so you have viable cell, you have total cell count and viable cell count there. And for bactericidal, you have total cell count, and then I'm just going to make a dashed line for for the viable cell count. That's going to go down. For bacteriolytic, you're going to have it go down. I'm not going to label it because that would take too much time, but so essentially what's happening here is, here you're inhibiting growth. Here, what's happening is the total cell number isn't changing, but the viable cell number is. So the cells are actually staying intact. They're not lysing. Whereas here, it's bacteriolytic, so the cells lyse. So that's why this total cell number decreases, because the cells actually rupture. So you can't count a cell that's ruptured. Okay, so MIC, that is the minimum inhibitory concentration. And that basically just tells you the lowest concentration of an agent that completely inhibits the growth of an organism. And you could test this by a serial tube dilution. So, yeah, it's serial. So tube dilution, and I'm pretty sure you guys did this in your lab, so you should be familiar with it. And the Kirby-Bauer disc susceptibility test, this is going to be, I don't know if you guys did it in your lab, but I did it when I took the class, and we just had like different antibodies or concentrations of antibodies on these little discs that you put in the media or on the plate, and then you grow it, and then you see which one has the biggest zone of inhibition. And you can measure that zone, and by that you could see how effective it is. However, that doesn't necessarily transfer perfectly to in vivo application. Because you could have like different concentrations or a multitude of other factors that you have in vivo versus when you're testing in vitro, which is a pretty controlled environment in comparison. All right, so the phenyl coefficient test. So that is what you use to rate disinfectants. And so with this, you're basically measuring bactericidal activity. So this is a bactericidal test. And So the disinfectants that are more effective than phenol are going to have a coefficient that's going to be greater than 1. And agents that are less effective have a coefficient less than 1. And then the two different methods were the Radio-Walker method and the Chick-Martin. So Radio-Walker just used water and Chick-Martin used 3% feces. So those are just the difference between those two specific tests for the phenol coefficient test. And let me see here. Okay. So distinguish the following chemotherapeutic agents. So antibiotics are naturally derived. So they're produced by like other microorganisms or other organisms to inhibit the growth of bacteria, such as penicillin is produced by fungus that's naturally derived. A synthetic agent would be something made in the lab. So that's not going to be like coming from a natural source. And an example of that would be like sulfonamides. And semi-synthetic would be like a combination of the two. So you could take penicillin and make a derivative of it, and it could have a broader range. Or you could also do that in order to prevent like antibiotic resistance by using the same antibiotic. Okay, so we're unfortunately running out of time, so I think I'm just going to cut it off for that review part. So if you have any questions, please ask. Does anyone have any questions? Anything regarding the exam or the review? I'm sorry, what was that? Oh, you want the answers online? Yeah, I could do that. I'll post that, I'll do that tonight. No problem. So for the answers, just go on my actual page for the SI. So just look up like UCF SARC SI on Google, and then you should come up with like molecular sciences, and then microbiology with Nicholas. Hope you guys know that's my name. Yeah, and then I'll just post the answers there. So, all right. Awesome. Well, thank you guys very much. I wish you the best of luck on the final. I don't think it should be too bad.